So with that, I'll introduce our speaker. Today we have David Nordsletten, who is Associate Professor in Biomedical Engineering and Cardiac Surgery.
Thanks, Marcy. It's a pleasure to be here with you all today, and it's interesting getting the invitation here to have a tech talk. Normally, that's not what we talk about. We usually try to hide a lot of the details. So I will talk a little bit about our work. So generally speaking, I'll introduce kind of what the lab is all about, and then I'm going to focus on the kind of key tools that we use and share some of those that might be interesting for you. So basically, sure that this is working. Let's see if this is, yeah, okay, cool. Yeah, so our lab is really interested in studying heart failure. And this is a big clinical challenge. And part of the challenge really with heart failure is that the disease is really complex. So the etiology of disease is very variable. And the disease really spans multiple scales, really. So you can see kind of features of disease all the way down at the sort of level of sarcomeres, the main units that cause your heart to contract, all the way up to the whole organ. So changes in how the shape of the heart looks and how it functions. And my team is really interested looking at, I'm going to drop using this, it's not working. My team is really interested in the sort of tissue to whole organ scale. This is kind of where we tend to work. And specifically what we're interested in is ways in which we can take medical imaging and computation and bring them together. And specifically, we're interested in biomechanics. So ways in which we can embed biomechanics, use it with imaging to better understand what's happening in the human heart. So what I wanted to talk about today is some of the tools that we use and have developed. And it's really on the side of imaging, image analysis, and modeling. And so I'm going to kind of talk through these three areas. So I'm going to talk a little bit about medical imaging and how we use that to visualize what's happening in the heart. Then I'll talk a little bit about machine learning. I am not a machine learning expert. I'll say that up front. I'm a modeler by training, but we do use machine learning increasingly more and more. And then I'll talk a little bit about biomechanics and how we leverage this. And I'm going to talk about it in two different contexts. One, which is sort of physics-based image analysis, and the other one, which is what we call patient-specific modeling. So looking at medical imaging, it's pretty amazing what you can do with medical imaging and what you can see. It really gives you a lot of perspective of what's going on and specifically in disease. And so one of the tools that we use a lot is magnetic resonance imaging. This is really a technique. I don't know how many people work with medical imaging data in the room. One. Okay, so a few of you. So apologies if it's simplified. But just to give everybody else an idea, basically, this technique uses magnetic fields to really probe what's happening in the body. So you sort of excite areas in the body. What you're exciting really is water molecules. You're basically giving them some excitation and watching the relaxation, and you can, from that, develop images of what is happening or what the heart actually looks like. So this is an example here. This is just a fly-through showing you the heart, and we can see it maybe in a second here. It'll loop back. But this is the torso here. Down here is the liver. And now we're coming back through. Here you'll start to see the heart coming through here. This is the left ventricle coming through here. And you can see actually, here's some of the great vessels going through. There's the aorta shooting down. So you can get a lot of detail about what the actual heart looks like within a patient from these images. The other nice thing is you can look at it over time. So you can use gated sequences for MRI where you get sort of movies that show exactly how the heart beats over time. And with MRI, there's a lot of versatility with how you excite in the body. And this gives you a large amount of versatility in what you're actually imaging. So another cool example that we use is what they call tag MRI. So this is 3D tagging, and what you can do is you can basically excite only in regions of the tissue, and you leave sort of unexcited bands, and then you are excited bands, and you can see those bands then move through time. So this is really interesting if you're looking at mechanics of the heart, because you can watch how the tissue is actually deforming and moving in space. So you can extract displacements. But there's a lot more that you can look at. And it's specifically in the heart, there's a bunch of different sequencers that you can leverage to start understanding disease. And I won't go through all of these, but this is just sort of an overview of what it looks like. So we have different ways in which you can look at relaxation. So how once you excite, the water molecules relax back to their sort of initial state. And so then these are sort of T1, T2 weighted sequences that can give you information about anatomy or fat or edema. There's something called late gadolinium enhancement, which is something we use a lot. This gives you an idea of regional fibrosis. So what you do is you give a contrast and you look at washout and you can see areas in where the heart is really not getting perfused right or has a lot of fibrotic buildup in the muscle. And then there's a huge number of other things that people are probing into as well. And there's PET MR as well. So you can really start to dig into what's happening in regions of the heart and see it in the anatomy. Another technique that we use in the lab is 4D flow MRI. So this is phase contrast imaging. And this is 3 different examples. So here we have the heart. This is a dissected aorta in the middle here. So what you're seeing, this little region here is where there's a tear in the aorta and it goes into false lumen. And this lumen here is the true lumen. And then over here we have phase contrast in the brain. So you can start to look at blood flow in 3D over time, and you can utilize this information to start better understanding what's happening to the cardiovascular function in these conditions. And we've been using this data a lot to try to help with diagnostics and risk. Another technique which we've been using in the lab is a technique called magnetic resonance elastography. And this uses the same kind of sequence as what I showed for flow, but instead what you do is you put a transducer on the body, which is represented by this little block here, and it vibrates. So similar to your cell phone, where you get this sort of like high frequency, low amplitude vibration that tells you someone's trying to ring you. We put these transducers onto the torso and you strap it down to them. It vibrates and sends waves through the body. And what you do is you establish some periodic steady state, and you can then image that with MRI. And so why would you do this? What's the point of that? Well, the relationship between how these waves move is directly related to the stiffness of the tissue. So it gives you a way in which you can try to probe what's the actual stiffness of that tissue in vivo without having to cut somebody open and actually poke at their tissues. And so this is a technique, it's predominantly used for liver, but it's been used a lot for cancer, breast imaging, brain imaging, and also some in the heart as well. In the heart, it's much more challenging, but it's something that people are trying.
So what's that technique called? I mean, what's the name of this?
This is MR elastography.
MR elastography.
Yeah,
That's good.
yeah.
Thanks.
Yeah. Okay, so the other technique or the other imaging that we use a lot in the lab is CT. And this is using a completely different technique. So instead of using magnetic fields to try to excite regions of the body, you're using X-ray and you're looking at attenuation as X-rays pass through the body. So the downside here is that you're sending X-rays to the body, whereas the other one doesn't have any X-rays that you're using. But the advantage is that you can get really detailed and anatomical pictures. So MRI to get the fine scale images like you could get in CT is possible, but the sequence takes a long time. So that, you know, the person will have to lay quietly in a scanner for a very long period of time to get this kind of resolution and clarity. But in CT, you can get it in seconds. So it's very fast. And you can get these beautiful images of the heart. It's a lot, you know, a lot of the times you use this for coronaries, or if you're looking really at the details of valves or anatomy of the aortic root. This is a common imaging technique that we use. But we can also, you know, get a lot of detailed information about the muscle of the heart and the anatomy, the shape of the ventricles, and also with CT, you can make them dynamic as well. So you can make movies showing how the heart's beating over time. And this is really helpful if you're trying to understand cardiac function. So you can look at these sort of movies of how the heart is beating. So these images are great. You're getting a lot of detail from the images. But the question is, how do you actually leverage that detail? So still, what we typically do clinically is just take those images and do fairly crude measurements from those images to get some idea. And those are actually, helping us to understand how to treat patients, but we're leaving a lot out, right? We're just sort of neglecting a lot of the detail that these images actually provide. And so one of the real tricks with this is how do we leverage it? And machine learning has really become a way in which we can do this really effectively. So that's kind of what I'll talk about. Machine learning, neural networks, AI. I don't know how many people do anything with that. AI, neural networks, raise hands.
Everybody.
Yeah, everybody. Yeah, it's everywhere, right? And in my world, in computational modeling and imaging, even for doing kind of reconstruction of images from raw data coming from these scanner systems, they're starting to use machine learning. So it's really coming up everywhere as a technique, and it's evolving fast, right? So there's a lot of different techniques that we can use, a lot of different new ideas that keep coming out and getting published. And my group, we're really focused mostly on how we can use machine learning for doing what I would say are interpolation tasks. So things that we can train a network to understand, and then it's just basically probing a very similar space again and again. So it's tasks that for us are very difficult, but for neural network algorithm, it's easy. So I'll just talk about a couple ways in which we do this. So the first one is on heart segmentation, okay? So what we get, as I said, is we get a lot of movies like this, where we see the heart beating over time. And what we'd like to be able to do is to actually extract the anatomy of this heart. So I want to be able to see where's the ventricles, these two parts here. Here's the septum, here's the other LV free wall. I want to be able to capture this anatomy and say how it moves over time. So the way that we've classically did this was very, very painstaking. Basically, you get a tool like this is ITK Snap, and you go in with a paintbrush and you fill in the areas of the muscle, right? So that's what we're doing here. And it's not that you do this once and you're done. You have to somehow make it dynamic over time. So either you have to try to use some sort of image analysis tool to say, how do I move this mask as it's going through time? Or you literally go in by yourself and every time step you go through and you segment again, right? I can say during my PhD, I actually did this every time step for a whole thing. And there we didn't have a nice tool. We had to do dots. We clicked dots into the image and we had point clouds, right? So this is something that, we talked about wanting to do modeling to leverage this information, but this is a big bottleneck, right? If somebody has to go in and do this. And so this is really where machine learning has helped us. So essentially what we use now is a UNet, which is using just a convolutional neural network. And we train this on about 10,000 segmented images. So some of these were segmented from other people and we pooled our resources together. So we have it from different scanners, different data sets, different diseases. And then we could train this UNet to give us these segmentations instantly, basically. So we can pass that information, pass the image in, and we get back these segmentations. Here we can see the ventricle in red, the ventricular wall in green, and then the right ventricle here in blue. We can't see the right ventricular wall because it's too thin, but we do get most of the segmentations coming from this. So similarly, another thing that we're interested in is finding valves in our images. So there's two clinical quantities. There's MAPS and TAPS. They're related to how valves move. And the reason this is important is because it tells us how well your heart can function during the contraction phase, where it's trying to pump blood to the lungs into the body, right? And so these two metrics are kind of a way in which you can tell if there's some sort of systolic dysfunction in the heart. And so what we wanted to be able to do is to actually identify these valve points and how they move over time. And of course, we could just do this. This is much simpler than doing the segmentation, right? Now I just need 2 dots per valve. But if you're wanting to do this automatically, this is going to be a painstaking process. If you have many, many images that you have to get through, this gets quite tedious. So again, we thought, okay, can we use neural networks to do this for us? And we trained this, again, we used a convolutional neural net, and we trained this on 8,500 segmented images. And really what we're looking at here is 10 landmarks. So we have different image types. So this thing that says 2CH, this is 2 chamber. You're seeing the ventricle and the left atria. And then you have 3 chamber and 4 chamber. So here you can see all 4 chambers of the heart. So we have these different views. They give us different views of the valves, right? So then in this case, we're seeing mitral valve. Here we can see mitral and tricuspid valve. So you're getting different perspectives on where the valves are, what they're doing. And what we wanted to do was to say, I'm going to give this neural network an image, and I wanted to figure out what valve it's looking at, where the valve is, and how that valve is moving over time, right? And actually, what you can see here, this is a tracking. It does a very good job of actually identifying this and identifying which valve it's actually looking at. So it can figure out from the image which one of these views it has, and it can track those points, the relevant points for that view. And then, of course, it's interesting to compare how well this works. And this is a little bit small, so I apologize. But basically, what we can see here are different plots for different valves or for different views. Here you can see 2 chamber, 3 chamber, 4 chamber. These are different views of the mitral valve. Then we have aortic valve and tricuspid. And this O here is some inter-observer variability. So what we had is 2 people actually, for a sample set, segment these valves and how they move. And we computed the error between their predictions. So how far apart were they? And then we compared with 2 image tracking techniques. So in that case, what we did is we set 2 points, and then we used image analysis to track those points over time to see where they would go. And we compared that with the neural network. And what you can see is that basically the observer error is on par with the network error. And the network, another nice thing is it didn't get anything to start with. So the image analysis techniques, we actually had to seed where the points should start. And that was something that we had to be very careful about, because if we put them into the blood pool, then they might just track to nowhere. So it really had to be pretty specific in order to get a good result with the image tracking. But with the neural net, it just knew where the points were from the beginning and would track those over time. So this actually is very effective for really finding these points efficiently. Okay, so the other thing that we've been using this for is for enhancing flow. So I told you guys about this phase contrast imaging data. One of the challenges that you have is, like any image, you have noise. You have, for flow, you have something called partial voluming effects, which basically give you some inaccuracies near sort of borders of your vessels or borders of the heart. And you can get aliasing as well, right? This is something where basically you exceed the maximum velocity that the image is allowing, and it will wrap. So it'll just go to the most negative and loop back, right? So you get this sort of wraps that appear in the phase of the image. And so what we wanted to be able to do was to clean this up. The other thing is that we're limited in the resolution, because you're trying to get these scans done quickly. You try to minimize the time, but that also means that usually we have voxels that are too big. And so what we said was, okay, maybe what we can do is we can train a network to look at low resolution images and give us higher resolution images. So it's a pretty simple idea. We basically took CFD simulations, and we generated artificial MRI data for these CFD images, or for these CFD simulations. And we rendered them at 1 millimeter and at 0.5 millimeter. And then we trained a neural network to basically look at 1 millimeter data and try to learn the physics in terms of how it could upsample that to a finer scale, right? And so from that, you can basically go through and you can train it to have a finer scale resolution here. These are just fly-throughs through a stack of this artificial CFD. And what's cool is you can apply this to then actual patient images. So this is not the heart, this is the brain, but we're using this in a number of different areas. And what you can see here kind of nicely is this upsampling. So this is an example of what the original resolution was in the brain scan. And then this is the upsampled resolution. And here again, you see the original scan. This is a little bit better resolution than here, but you can get even higher as you go to this low res with the super resolution. And what we've seen, which I'm not showing you here, but what we've seen is the things that we're interested in calculating from these flows are more accurate when you use this kind of upsampling and filtering that you get from super resolving. So again, it's another area in which neural networks are kind of helping us to figure out a smart way in terms of how do we interpret these images and get a better quality image in the end. Okay, so I'm going to talk a little bit then about how we use this stuff. So we have images, we have ways in which we can extract information from these images using machine learning. And now what do we do with all of that? So there's sort of, like I said, our lab is interested in biomechanics generally. And there's sort of two ways that we look at this. One way is to say, can we just take the images and use biomechanics ideas to directly infer something from an image? So then we're not really modeling anything per se, we're just using physics principles. The other way is to say that we're going to actually try to simulate what's happening in vivo using computational techniques like finite elements, okay? So I'm gonna first just quickly talk about the physics-based image analysis, and then I'll jump into how we do modeling. So the big thing that we're doing here, there's a number of different areas that we're doing this, but I'll just talk about one. So the one that I'll talk about is about using phase contrast images. And really what you're interested in when you're looking at phase contrast images, the most important, I would say, clinical tool that you can get from this is what is the drop in pressure that's required to get the flow that you see. This is used in a lot of different cardiovascular conditions to determine whether or not somebody should go for a procedure. So for example, if you have a valve stenosis, so your aortic valve, for example, if it can't open properly, it leads to a constriction that blood has to go through. And this adds to the work that the heart has to do. So it increases the amount of pressure the heart has to generate in order to drive blood flow. And so if you can look at the drop in pressure, that gives you an idea of how severe is that disease, right? And so what we'd like to be able to do is to actually derive that pressure drop from images. And you can do this this is something that's been done in the case of aortic stenosis a lot with echo, because echo is another imaging technique. You can put a probe on the body and you can quickly get images. But echo can't get everywhere, right? So it needs to be somewhere that is easy for the probe to actually see. And the aortic valve is one that's fairly easy for the probe to see. But if we want to think more broadly of cardiovascular conditions, then we need a tool of how we could first look at flow throughout the whole body, and then a way of extracting what that flow means in terms of pressure. And that's what we've done here. So this is a simple formula up here. What we've done, I'm not going into all the details, but basically what we've done is we've taken Navier-Stokes equations, which governs fluid flow for a Newtonian fluid, and we treat blood at this scale as Newtonian. And what you can do is you can sort of rearrange that equation to get a simple scalar relationship between a drop in pressure and different quantities that you can calculate directly from the flow field. So looking at changes in kinetic energy, how much blood you're actually ejecting through the system, and then any sort of viscous dissipation that might be occurring. And so what this plot here shows is the correlation between our estimates using this technique we call V-work. It's a virtual work energy relative pressure. That's what this formula is. And the actual invasive catheter measurements that were taken. And you can see that this correlation shows a nice kind of relationship between the non-invasive measure and the measure that is acquired, basically sticking a catheter in somebody and measuring pressure directly.
Wow, that's great. The idea you're trying to get away from putting in the catheter.
Exactly. Yeah. Trying to avoid that if possible. Another cool thing that we've been doing in this space is looking at flows that go turbulent. So turbulence is basically where flow kind of breaks down. Usually what happens is when flow breaks down, it amps up the amount of pressure that's required. All right. So in severe disease, usually you'll get some development of turbulent blood flow where it's just very chaotic. And what's cool about MRI is not only can you sample the velocity, you can sample it multiple times. So what you can do from an MRI image is you can get the flow and also the covariance in the flow. So how much variability do you see? If I measure the same point at the same time in the cardiac cycle, do I get the same value, right? Or if I don't, how much does it vary? And that gives you flow covariance, which is directly related to turbulence. And so again, you can basically image this covariance, and if you plug it into this formula, you can again recover a nice relationship between pressure drop and the flow. And we've been, I showed an example of dissection in one of the slides, and we've been doing this for dissection as well. This is actually a phantom that was developed at Stanford. It's a really beautiful model. But what we're looking at here is an aortic phantom where it has a dissection. So there's a little tear that's up here. And then it has a re-entry tear, what we call re-entry tear at the bottom. So you can have flow that goes through one channel that's kind of outside here, or in this bigger channel that kind of wraps behind the small channel here, and they reconnect at the bottom. So these two images up here are showing velocity measurements that were taken within the MRI. So this is a magnitude of the velocity field, so you can see what's happening in the flow. And then these are actual maps of pressure that we derive directly from this data. So again, using these techniques, we can basically come up with pressure maps. And what's really interesting here is showing how changes in the morphology of how these entry and exit tears occur in the model changes the pressure that you see within this artificial chamber that's torn open, right? And this is really important because what we think is happening in these diseases where you get a dissection and you get a rupture, which is the worst outcome that you can get. That basically means your aorta ruptured in the body and you're bleeding out into your chest. We think that this is largely occurring when you have abnormal pressurization in that additional chamber, what we call false loop. And you can actually see this here. We just changed in these two models the size of the exit tear, and it basically fundamentally changed how much pressure gets generated in that whole chamber. All right. So next I want to talk about how we can use these things for patient-specific modeling and the tools that we use there. So in this case, what we're trying to do is we're trying to basically use image data together with some of these metrics we can extract from machine learning and recreate what's happening in the heart. And we do this in two different ways. I'm going to talk about biomechanics and fluid mechanics. Okay, so these are two different areas that we use this. So I'm going to walk through how we do this in our pipeline. So basically what we get is those images that I showed you earlier of the heart, and usually you get them in 2D. So you have to first figure out how they all align in space. And so we use a registration toolkit in order to get all those images to really align well with each other. And then after that, we can use this neural network that I showed you before to get segmentations of the heart. So again, now we have the left ventricle, the blood pool, and the right ventricle. And then from that, we can also track the valve motion. So we can see exactly where the valves are moving over time. So it gives us an idea of where the ventricular chambers are moving relative to the atria. And from this, then we can actually create a computational model, like an anatomical version of that heart, right? And what we use for this is a diffeomorphic mapping algorithm. And so essentially we take a template heart, we take these segmentations and we morph them to match each other, right? So we morph the template heart into the patient heart that we observe. And this is actually really helpful because it helps with a lot of the artifacts that you get in images. So these are examples of some of the images that we get. I showed you first the nice clean ones, but you don't always get clean images, right? So here you can see we're getting a big artifact that's streaking across the heart. Here it's quite noisy. And the neural network does a decent job of actually still extracting where the heart is. But you can see here, for example, there's a little hole in the middle of the heart there. And we don't want to have to always go in and manually adjust these things, right? So a way to get around this problem is to actually use template models that try to fit the data, but have some inherent restrictions to them. In this case, it's that it has to stay a heart, right? It can't all of a sudden be a heart with a hole unless that's our template model, right? So basically we can end up getting a good recreation of this geometry, even though there's some errors in the segmentation. Okay, Another important thing with the heart is a muscle, right? Just like your muscles in your body, we know that the muscle acts in a specific direction, right? If you contract your bicep, it's going to pull your arm up. Your heart is also a muscle, and it also has a directionality to it. And that's really important for how it actually pumps. And in the heart, you get this complex, what we call fiber structure, right? So it's telling you how the muscle is oriented throughout the whole tissue of the heart. And we need to be able to recreate that in order to represent what's happening in the images. And so what you can do is you can use rule-based methods for basically taking this anatomy and generating what the fiber field looks like based on some idea of what it should be. And this idea of what it should be is based on DTI data. So again, it's another MRI technique where you can look at diffusion tensor imaging to understand exactly what the fiber structure is throughout the whole heart. And what we've done recently is to look at not only the ventricles, but also or not only the left ventricle, but the right ventricle and up to the valve. So looking at exactly what does this anatomy look like and how does it compare with these DTI images and the actual angles of fibers, muscle fibers within these hearts. Right, so that we can get all of this data from MRI and we can actually basically come up with good rules that recapture that sort of structure and embed that in our hearts. All right, so with the anatomy, we can come up with boundary conditions, and from the valves, we can also create how the valves move. We can plug that into physics-based models and run basic physics on this heart to make it contract. And here what we're solving is simple F equals ma for a whole heart, right? So we're solving Cauchy's equation of motion. So what does that look like? So the finite element problem that we end up solving for people who like finite elements is this, and it's quite long, but it incorporates everything that we have here. So we have basically a PDE that governs how stress balance occurs within the muscle. We have constraints on exactly how the valves should move. We have pressure that's exerted on the heart walls itself, and these can either come from data that we're given, or it can come from what we call 0D models. So these are simple models that recreate the cardiovascular system and how pressure and volume are interrelated. And then we have constraints that try to basically keep the heart in place as we see it in the images, and constrain the volumes of the chambers as well to match what we see in the image data. So the nice thing about this setup, the way that we have it, is that we can assimilate data in one go as we simulate. So a lot of times what tends to be done in patient-specific modeling is you do some iterative process where you try to run the model, look at its results, correct it, run it again, see how it looks to the data, correct it, and you just go through in an iterative process. But here we can actually do one simulation where we are solving for some of these parameters that are unknown as we step forward in time. And so what this gives us is nice simulations like this of cardiac function. So here we have the right ventricle in red and the right ventricle in blue and the left ventricle in red. You can see these heartbeats. And what's cool is you can then start to look at things that you can't see in images, like the active stress that's being generated within that muscle to get that heart function that you see, and plot that over space and time. You can also look at strains in different regions of the heart and how these are varying in different diseases. So it gives you some idea of function and how it's kind of comparable between healthy and diseased hearts. And you can also start to look at things like fibrosis. So this is something where we can use fibrosis imaging and embed that into this computational pipeline and basically try to account for the physics that are happening there. So the nice thing is once you have this personalized heart, you can start to change things and see how it would interface or change how the heart actually beats. So you can do sort of virtual procedures on these hearts to try to look at how they might be able to predict how that patient's heart will respond. And this is something that we're also interested in and working on. Okay, so I'll also touch base on how we do this for hemodynamics modeling. So stepping away from solid mechanics, and we'll look at fluid mechanics. And the process is very similar. In the case of fluids, we often are starting with CT data. And again, in this case, we have a neural network that we trained on 3,000 CTs to give us segmentations. In this case, we get the ventricles and also the atria and the great vessels. So you get a few other different compartments that you can kind of get automatically. Nice thing again is that you can apply this to a dynamic CT to see the anatomy, or you can apply it to a static CT or a dynamic CT. So you can either see just the anatomy or the anatomy and how it changes over time. And once you have that, you can basically create a computational mesh. This is a mesh of the left heart. So you have the left ventricle here, the left atria, and this is the aortic outflow tract. And then you can use, what we use is image tracking to basically take these segmentations over time and morph this mesh to match what you see in the images, right? So then we have a moving domain that moves exactly like the heart from the images. And then we can start to look at solving blood flow through that. And again, the nice thing about using this with the neural net, if we tried to do this by hand, it would be a disaster. So the CT images are very, very fine. So it would, the number of slices that you have to go through if you were going to do something like painting would be just disastrous, right? But with neural nets, you can get these segmentations in hearts very quickly. And then you can throw these into tracking algorithms and then get these morphing models of how the heart beats and how the chambers move with much more ease than we could before. All right, so then once we have this, we can plug this left heart into some sort of lump parameter circulatory model, again, representing the cardiovascular system. So in this case, what we do is we have our ventricle, which has an aortic outflow, and that plugs into a systemic Windkessel model. And that Windkessel model goes then through capillaries into veins, pops back to the right heart. And we have, in this case, a right heart lumped parameter model. And then that goes through to the pulmonary system and then loops all the way back into the left atrium. So the nice thing about this is you have a closed loop system that's basically able to kind of give you how cardiovascular function will change and will feed back through the whole cycle. So through the whole system. So if you change output, that's going to eventually circulate through, and it's going to come back into the input that you get into the left atria, which is what happens physiologically in our bodies, right? And so we can utilize these to run simulations of blood flow. This is a case of blood flow and mitral regurgitation. So what you're seeing here is this is actually maps of vorticity. So it kind of gives you a better idea of what the flow is doing. But what happens here is you get inflow, and then you're getting regurgitant jets that form in the left atria. And that jet is caused by the fact that the valve that's supposed to close between those two chambers is not closing. So it's leaving a gap and allowing blood to flow backwards. And why this is a problem is basically it's fighting the other side of the heart. So you have the left heart pumping blood to the body, but also pumping blood against the right heart, which is trying to pump blood to the left heart, right? So this is a big problem, but we don't really understand how these hemodynamics really relate to the risk of impact on the right ventricle. So how does that maybe cause the right ventricle to fail? And that's kind of what we're trying to study with these computational models. Another case we're using this for is looking at TAVR. So this is transcatheter aortic valve replacement. So in this case, sort of a new technique. Well, it's not too new. It's been around for a while. But basically, it's a technique where you can implant a new valve into a patient without having to open them up. So you use a catheter to put the valve in place, and then you expand the valve, and then you can see, or you can fix the valve that might be dysfunctional, either through insufficiency, where it basically allows blood to go backwards, or where it's stenotic, so it doesn't allow blood to flow out. And what we're interested in here is really how do we get the best outcomes for patients, and how do we make sure that there's a good long-term plan. So what they've been seeing with these TAVR valves is that in patients, they're often living much longer than was expected. And these valves, they're hitting the end of life for these valves, where the valves are now starting to become regurgitant themselves, right? And the challenge is that you put in this valve, which is a big stent with a valve in it, and now you have that valve not working, right? And you can't easily go in and rip that stent out because it basically gets grown into the tissue.
Yeah.
And you need to figure out what you're going to do, right? And so what we're trying to think of is, when you're doing this procedure, how do you think long-term, right? Knowing that you're going to maybe have to replace this, and what's your strategy for doing that, right? Because if you look at images of this, it's kind of nasty. When they do this, what they call a tab in tab, they put a new tab inside this original one. It's messy. Right?
How long do they last, the first one?
So they're supposed to last 10 years, but it's 5 to 10. Yeah. So that's
heavy duty.
yeah, exactly.
Putting a number on it, I mean, because wow.
Yeah, exactly. Yeah. Okay, so that was pretty much all I wanted to go through, walking through kind of what we do, how we use imaging, machine learning, and biomechanics to start looking at the heart. And this is just my group who does all the work that I talk about. And I'm happy to take any questions that people have.
As a reminder for those who are online, if you have any questions, you can put them in chat or raise your Zoom chat hand or Zoom hand, or we can call on you to unmute. Okay, we're in the room of questions. So do that.
Just 2 questions first. The first one is, because I don't have experience with the cardiac imaging, my experience is confined in neural imaging. So when we place patients in the MR scanner, usually they have to stay still to avoid any motion. But heart beats all the time. So how can you make sure you can get quality images when the heart continues to beat?
Yeah. So what we usually do when you're doing cardiac imaging, there's 2 ways that you go about it. Depends on which kind of scan you're doing and how long it is. So one way is that you have 2 different sources of motion, right? You have the heart motion, but you also have breathing, right? Which is changing where the heart is. So your diaphragm goes up and down, changes where your heart is. So if you image in a plane, what you're actually imaging is changing if the heart is shifting with breathing. So one way that it's done is to do breath holds. And then what you're doing is you have an ECG that's measuring the heart cycle. And then you can basically trigger and bin data according to where in the heart cycle it occurred. So if you hold your breath, then you can sort of trigger and get bins of data that you can reconstruct the heart cycle with. The problem is you can really only get like 1 slice in a breath hold, right? And if you want to have 15 slices, like I was showing there, that's typically what you get when you get a stack through the heart. You have to do multiple breath holds, right? And the problem is that you're now relying that every time the person is going to hold their breath exactly the same, which is why we have to do this registration, because it's never perfect, right? The other way is using free breathing. And there, what you typically do is you use a navigator. So what you'll do is you'll have a very low weight, quick sequence that pulses a line through the diaphragm, and you look at where the diaphragm is. And you only allow collecting data if the diaphragm is in a certain window, right? So then the patient can sit there and breathe. And then if they fall into the window, you acquire data. And then you try to figure out, you also have the ECG. So you have to try to figure out where did that data, where should it go? Which bin does it belong to? And that's takes longer because it depends on how well you set this up, right? And how well the patient breathes. So if you're a good breather, you hold a position for a long time and then you do an exhale, inhale, and then you hold the position for a while. But sometimes you'll get people who breathe really weird. Like they have this sort of sawtooth pattern and you're like, you only acquire data for like a brief snapshot in time. So the efficiency that you have can be a variable, and that can change how long these scans go. Which is, what you have to do to get cardiac.
That sounds good. And the second question is about segmentation. So usually, again, back to neural imaging, we have this template for segmentation. So it will make the whole segmentation much easier. But it seems that in cardiac, I wonder if because there's no like a reliable template so far, that's why you had to develop like with the neural machine learning to do the segmentation.
So I'll say that, so the CT1 that we're using is actually based on a template that it tries to fit. It doesn't fit a template, but it basically, the data that it's using as training data was based on a template. So where it did template fitting, so it had an anatomy that it would morph into place. But for the MRI, no, we don't have a template, and it's mainly because of the views that we get. You know, where you get these stacks of images, it's not always reliable, and sometimes, especially near the base of the heart, you'll have images where you'll see the whole ring of the ventricle, for example, and then because the ventricle moves longitudinally, it moves up and down, it sort of will pull in the valve. So all of a sudden the myocardium will disappear, but you still want to segment that image. So you have this sort of partial crescent that you're supposed to try to recreate. So doing a template doesn't work as well in that case. We don't have 3D data like you'd have in the brain. So in that case, it's easier to use these straight up neural nets and then use a template fit at the end to basically say what should the anatomy look like based on the segmenting that I see from the neural net.
If I may, one final question.
Actually, Chris, let's hold up. There's
Yeah,
actually a
sure.
question online. I want to make sure we're giving equal opportunity to everyone, and then you can get back here. So Dana online asked, great talk, thank you. A question about the CNN training for segmentation. Are there any known or potential variation in heart vectors that correlate with patient demographics such as sex, BMI, race? And how is your training set evaluated for possible or unintended biases of the physician?
That's a great question. And I'll say that we didn't do anything to look at that. We kind of, in this case, we had data and we used the data that we had. So we didn't have, you know, a choice of 3,000 or, you know, up to 10,000 segmented images that covered all demographics. So we just had the ones that we had, which isn't a great answer, but it's the true answer. I don't know how it biases. I've seen this done actually, looking at how some of these neural net segmentations are biased. There was a paper, I can't remember off the top of my head, that was about this specifically. And it did show that there was a bias. But I don't know in our case what that looks like. Certainly there is variation in anatomy. And that seems to be okay. So one of the big areas you see this the most is in the left atria. How many veins, pulmonary veins, come into the left atria varies between all of us, right? So it's not like you're going to always see the same morphology. Whereas the ventricles they're going to be, you're going to have, unless you have a congenital condition where you have a single ventricle, 99% of us will have two ventricles, and they'll be kind of the same shape, they'll be wrapped in the same way. So there's a similarity. So you can imagine that you could easily warp a shape into that to make it match. But with the atria, it's not as easy because you have fundamentally different topology to the structure than what you, from one patient to the next potentially. So there is, it seems to be able to cope with that relatively well. But again, I don't know about the bias for gender or race or any of those things. Yeah.
Thanks for this wonderful talk. It's absolutely great. My question is about, you know, the economics of this from a personalized medicine perspective.
Yeah.
You know, now I think one thing we have going for us is that these procedures are generally pretty costly anyway. So, you know, but just in terms of rolling it out there so that people can equitably have access to the personalized, you know, modeling of the heart,
Yeah.
you know, going forward.
Yeah.
You know, how much would this cost to do, and who could have access to it, and what's the potential cost savings that would offset the cost of doing it?
Yeah, these are great questions and really important to think about as you develop technology, right? Where are you going to get the impact clinically? I would say there's multiple answers, and it kind of depends on what you're trying to do.
Yeah.
So I'll say one area that we're maybe closer to doing something clinically, and then I'll talk maybe about the modeling. So one area where we're utilizing this, or we're trying to utilize this is in aortic dissection. I showed you guys some of that data, right, where you get this dissected aorta. A challenge that we have at the moment is how do you predict this risk of rupture? So is the patient's aorta going to rupture imminently? And so you should do something, or is it going to be stable and can you wait for a while? The reason why you might want to wait is because there's other complications to the procedures that you do to try to intervene, right? So if you don't have to intervene, then it's kind of better just to let it be. And so there we're trying to use this medical imaging data. We've shown that some of these relative pressure measurements seem to correlate with the risk of growth of that new chamber that's sort of torn open on the aorta, this false lumen. And that growth rate is a real predictor of rupture risk, right? If the thing keeps ballooning, then you know that this is maybe closer to rupturing. And so the idea here is we don't have anything right now. We already get a lot of imaging for these patients anyway. So if what you had to do was to put them in an MRI scanner and you could get an answer about their risk over the next year or two, then this would be better than what we're doing currently. Because right now we just look at the diameter, that's the best correlate we have, but it's weakly correlated. It's not very good. So that's something where I think the cost would be minimal. How we treat the patients, it wouldn't be a big change to what they're doing. And so the bar would be pretty low, right? Of course, you'd need to have a CT and an MRI scanner. So, if we're talking about, maybe other countries that don't have as much access to these systems, that's something we can't really help.
But in a place like this,
yeah,
it can be done into the workflow.
Yeah. On the biomechanical modeling, the patient-specific modeling, this is really more kind of longer term vision of how this could come in. It's not something I would say is ready to be implemented right now. There's a lot of things in these pipelines that I showed you that are still not automated, that we need to come in. For example, I say we get the segmentation, but the mesh, we have to make the mesh, right? So somebody has to go in and make it. It's not like we can get this directly. So there's a lot of steps in that process that need to be refined. We need to understand the uncertainties, how that impacts the actual results. But the idea would be to essentially try to wrap these up. And two ways you could do that. One is to try to automate the process. The other way is to try to use model order reduction on these to basically do a more direct link between the image and an output that we care about clinically, because maybe we don't need to have all of the physics to answer an important physics question about what's happening. And so that's another route that we're looking at in terms of utilizing these tools. One area that is an exception to this is if you're looking at response to maybe a treatment, like surgical intervention. So for example, I showed you that video of flow with a mitral stenosis, where you had sort of blood flowing backwards in the heart the wrong way. So what we can do in that case is we can do sort of virtual clipping. This is a technique that they use to try to solve this problem. And we can look at how that would impact the hemodynamics, right? So this is a pretty easy thing that's, again, we don't need any other data than what's getting collected clinically anyway. And we could basically give a surgeon an idea of where should you put the clip and how many clips are you going to need to really stop the regurgitant jet that you're seeing in the patient. And this is something that we're working on at the moment. I think, again, that's a low energy, but just the broad biomechanical model where it has everything you want and you can poke it in every which way and it works. I think that's still a ways away for being used clinically.
Yeah, but I mean, being so old as to going to classes way back in graduate school before there was MRI, we were still calling it NMR, and the first 3D systems weren't even built. We were talking about building. And then the ITK, you mentioned that. I was involved in helping to develop that with Bill Lorenz and those kind of people way back in the day. Fast forward to now, I mean, what will happen in 20 years will be quite amazing. And I would predict that a lot of it, the meshing, you'll figure out how to do. All this is going to fall. And it's going to be a consumable on the back end of the system that's going to be hooked to the scanner.
Yeah.
This is where this is going.
Absolutely. Yeah. I mean, the medical imaging companies already have teams that are working on these things. And I think their goal will be to either develop it in-house or buy a company that's doing it.
Yeah.
That's usually what. So we have a project on the elastography that we've been we've done some work with Siemens on that, and they basically told us, like, yeah, that sounds great, make a company and we'll buy
Spin it off and they'll buy you off.
Yeah, exactly. Yeah. So yeah, this is, I think, where they're going as well, is to try to, as much as possible, have it on the console. When you get the image, you just say, I want this assessment report, and then it just runs it for you.
Yeah. So I would say, given who we're talking about, Siemens, General Electric, so on. 15 years.
So,
It'll
I
be
think
there.
they'll be much closer, if not already in there.
Yeah, 15 years.
And then maybe you could even do the virtual procedures on the scanner too. That would be super cool.
I could definitely see that.
Yeah, that would be a big sale.
So it's, yeah.
It's
So your
gonna-
challenge,
things
what
are
are you gonna do next?
Well, I've got a few years.
That's
Figure it out.
awesome, yeah, it's really interesting.
Did you have a question you wanted to ask?
We're good.
Okay. My question is about DTI.
Yeah.
Because usually DTI, the resolution is lower than T1. So I wonder if you want to like be more sure about the anatomy from DTI, would you usually like co-register to the T1 or C?
Yeah, so we usually co-register with like a SSFP image
Yeah,
to get that. So it is lower res. So there's two ways that we are kind of working with DTI. One is with ex vivo hearts. The nice thing there is that you can basically put the heart in a bucket and you can leave for 12 hours, come back, and you can scan to your heart's desire, right? So you can get really fine resolution images from that. Obviously, you can't do that in vivo, right? So in vivo, there's totally different scans. Usually, instead of getting a whole heart and getting DTI images through the whole heart, you're getting 3 slices, and they're coarse, right? And so you're getting only 3 slices, and that still takes quite a long time.
Wow.
But this is also kind of a little bit amazing that they can get data that is coherent, right? So you're trying to measure diffusion in a heart that's beating and moving in time, right? The heart is actually moving much more than the diffusion that you're trying to image, right? So it's a lot of tricks that they need to do in the sequence development to make that actually work. But the results look coherent. I mean, it doesn't look like it's totally nonsense. So, yeah, but it is in vivo. It's a challenge. You're limited to the data, and we have to do registration with other higher resolutions. Makes sense.
Thank you for the talk. It was really interesting. For many applications, not just predictive accuracy is the most important, but the uncertainty quantification.
Mhm.
And I'm curious if you're wrapping like conformal prediction around some of these model outputs, or ways that you're saying we're going to fall back to manual segmentation and or alternative strategies, or collect more data, or other
Yeah.
sort of decision points that may be important.
Yeah,
Good.
we need to so there's 2 answers, right? So the one is on the sort of physics-based image analysis. There, it's a little bit easier because what we've been doing is essentially a sensitivity analysis. So we look at altering the segmentation and how does it change the output and how much variation. The other thing that we do is we add noise to the images, and we look at how much, so you can get an estimate of what the SNR is, the signal to noise in the image. And then you can basically pollute your image with the same SNR, and you can see how much it changes your result, right? So that gives you an idea of how much confidence you have. And those are pretty good to give you kind of an understanding of how much you can sway what you're predicting. But on the patient-specific modeling side, I mean, that pipeline, there's a lot of different places where you're kind of pumping in data, and that could maybe change what your outputs are. And I think, we've gotten to the point where we have pipelines that work pretty reliably, but we need to kind of go through and start to look more at the uncertainty. So there we haven't done as much. We still do some basic things like changing the segmentation a little bit, but that's not the only thing that we're plugging into the model. There's a lot of data that's going in. So we need to dig a little bit more into that, but we haven't. There's people in the community who have looked at this, but we haven't started yet on that front.
I think we are at the hour, so we should probably wrap up. We can thank David again.
That was really something, man.
Thanks.
Good work.